Mutations are changes in the genetic sequence, and they are a main cause of diversity among organisms. These changes occur at many different levels, and they can have widely differing consequences. In biological systems that are capable of reproduction, we must first focus on whether they are heritable. Specifically, some mutations only affect the individual that carries them, and while others affect all of the carrier's offspring and further descendants. For mutations to affect an organism, descendants must 1. Occur in cells that produce the next generation, 2. Affect the hereditary material. Ultimately, the interplay between inherited mutations and environmental pressure generates diversity among species. Mutation rates can vary within a genome between genomes. Because mutation is one of the fundamental forces of evolution, such work will continue to be of paramount importance. Changes in chromosome number can occur by aneuploidy and euploidy. Aneuploidy refers to a variation in total chromosome number in a cell or organism by addition or deletion of chromosomes. In fact, aneuploidy does not change the number of chromosome sets. It only changes the normal total number of chromosomes in a cell or organism. This variation affects the genetic balance of the cell or organism. It changes the amount of genetic information or products of a cell or organism in an abnormal condition, which can lead to different syndromes such as Down syndrome, Edwards syndrome, triple X syndrome, Klinefelter syndrome, Turner syndrome, and Cre-Duchat syndrome. Monosomy and trisomy are two common aneuploidy conditions seen in an organism. The term monosomy is used to describe a chromosomal abnormality in which one chromosome is absent from one pair of homologous chromosomes. The term trisomy is used to describe the abnormal chromosome number in which three chromosomes are present from one type of homologous chromosomes. Those two conditions can be indicated as 2n-1 and 2n plus 1 respectively. There are another two types of aneuploidy conditions named nullisomy and tetrasomy. Nullisomy refers to the abnormal chromosomal composition that occurs due to the loss of both chromosomes in a homologous chromosome pair. It can be indicated as 2n-2. Tetrasomy refers to the abnormal condition that occurs due to the addition of an extra pair of homologous chromosomes, and it can be indicated as 2n plus 2. All these conditions cause abnormal chromosomal numbers or numerical changes in the total number of chromosomes. Euploidy refers to the variation in the complete set of chromosomes in a cell or organism. It is common in plants and occurs in high frequency than animals. Since chromosomal number in a cell affects the sex balance of animals, euploidy in animal cells results in sterility. Hence, euploidy is often related to plants more than animals. During euploidy, the entire set of chromosomes is duplicated once or several times during cell division. Diploidy, triploidy, tetraploidy, pentaploidy, polyploidy, autopolyploidy, and allopolyploidy are different types of euploidy conditions. Comparison of euploidy and aneuploidy. Euploidy is a variation of a chromosomal set of a cell or organism, while aneuploidy is a variation in total chromosome number of a cell or organism. In number of chromosome sets, in euploidy, the number of chromosome set is changed, and in aneuploidy, the number of chromosome set is not changed. In chromosomal composition, in euploidy, cells have states of 3n, 4n, and etc. In aneuploidy, cells are states on 2n plus 1, 2n minus 1, n minus 1, n plus 1, and etc. Euploidy occurs due to fertilization of one ovum with two sperm, etc. And aneuploidy arises due to non-disjunction in meiosis 1 and 2 and mitosis. Euploidy is not seen in humans, while aneuploidy is seen in humans.
Hi everyone, previously we covered about changes in chromosome structure, and now we will go In gene mutation, we have microlesion base pair substitution, frame shift mutation, mutator genes, and transposon or jumping gene. Microlesion base pair substitution is a replacement of one nucleotide and its partner in the complementary DNA strand with another pair of nucleotides. There are three different results that are possible: silent mutation, missense mutation, and nonsense mutation. First is silent mutation. In silent mutation, a change in one base pair has no effect on the protein produced by the gene. This is allowed for by the redundancy in the genetic code. In the following example, both GGC and GGU code for the amino acid glycine. Then, the mutation is silent because it causes no change in the final protein product. Second possible result is a missense mutation. In a missense mutation, a change in one base pair causes a single amino acid to be changed in the resulting protein. The result is called missense since the code is now different. In the following example, GGC has been changed to AGC, resulting in a different amino acid. The effect of a missense mutation on the protein is unpredictable. A missense mutation is the cause of the disease sickle cell anemia. The effects of a change in one base pair alters one amino acid in the resulting hemoglobin protein, causing red blood cells to take on a strange shape. The strange shaped red blood cells cause a variety of phenotypic effects in the individual. This is an example of pleiotropy. A missense mutation is not always bad. It could create a protein that improves reproductive success or survivability. Natural selection could then help increase the frequency of this new allele in the population. Third possible result is a nonsense mutation. In a nonsense mutation, a change in a single base pair creates a stop codon. Because this kind of mutation creates a stop signal in the middle of the normally functional gene, the resulting protein is almost always non-functional, hence the term nonsense mutation. In the following example, UAG instead of AAG makes the codon stop. Let's move on to frameshift mutation. Frameshift mutations involve the addition or deletion of base pair, causing a shift in the reading frame of the gene. This causes a reading frame shift, and all of the codons and all of the amino acids after the mutation are usually wrong. Since the addition of amino acid to the protein chain is determined by the three base codons, when the overall sequence of the gene is altered, the amino acid sequence may be altered as well. Example, this is the original DNA strand. We'll make an insertion of an adenine base between the Gs. Also, below is the resulting mRNA strand, and here's the resulting polypeptide: methionine, leucine, isoleucine, serine, and another leucine. If this kind of mutation happened, then it would turn out very strange. It would be incorrectly synthesized due to serious defect in the amino acid chain. Let's go to mutator genes. A gene that increases the spontaneous mutation rate when it is mutated is called the mutator gene. Wild-type mutator gene products are involved in DNA replication and repair. The most popular example of mutator gene known in corn relates to a gene which is located on chromosome 3 and causes absence of anthocyanin, leading to loss of purple color in corn kernel endosperm and other parts of the plant. This mutation can occur even in somatic tissues, leading to spots in kernel allurone layer, figure on the right side, or stripes in stalks and leaves. Since the kernel allurone is triploid, the effects of as many as 3 doses of DT can be observed on mutability of A, as shown in table 21.8, or figure in the left side. On the left side. Let's go to transposon or jumping genes. Transposons are segments of DNA that can move around to different positions in the genome of a single cell. In this process, they may cause mutations. These mobile segments of DNA are sometimes called jumping genes. Presence of transposable elements was first predicted by Barbara McClintock and Korn in late 1940s. After several careful studies, she found that certain genetic elements were moving from one site to an entirely different site in the chromosome. She called this phenomenon changing site of genetic elements as transposition, and those genetic elements were called by her as controlling elements. These controlling elements were later on called as transposable elements by Alexander Brink. In late 1960s, this phenomenon was also discovered in bacteria. Different transposons may change their site by following different transposition mechanism. One is cut and paste transposons, two replicative transposons, and three retro elements. Let's start with cut and paste transposon. They transpose by excision or cutting of the transposable sequence from one position in the genome and its insertion or pasting to another position within the genome. The cut and paste transposition involves transposase subunits. Each transposase subunit binds to the specific sequence at the two ends of transposon. These subunits of transposase protein then come together and lead to the excision of transposon. Let's go to replicative transposons. They transpose by a mechanism which involves replication of transposable sequence, and this copy of DNA so formed is inserted into the target site while the donor site remains unchanged. Thus, in this example of transposition, there is a gain of one copy of transposon, and both the donor and the recipient DNA molecule are having 1-1 transposable sequence each after transposition. Retro elements. Their transposition is accomplished through a process which involves the synthesis of DNA by reverse transcription by using elements RNA as the template. This type of transposition involves an RNA intermediate. The transposable DNA is transcribed to produce an RNA molecule. Let's go to reverse mutation. Reverse mutation or reversion is a production by further mutation of a permutation gene from a mutant gene. Reversion is the correction of a mutation. It occurs at the same site. More loosely though, the term is applied also to a mutation at another site that masks or suppresses the effect of the first mutation. In fact, such organisms are not non-mutant but are double mutants with the same phenotype. A reversion mutation typically would be a kind of beneficial mutation, or at least that is the case if the original mutation is or was a detrimental mutation. A reversion mutation, in other words, can be one that restores a beneficial allele, though strictly speaking only restores is necessary for mutation to be reversion mutation.
So that is gene mutation and reverse mutation. I am Niti Lagunday. A while ago, we tackled about gene mutation. So now I'm here to discuss about mutagenic agents. So now we're going to talk about mutagens and how they can cause DNA damage. First, what is a mutagen? Well, a mutagen is any chemical substance or physical event that can cause genetic mutations. Chemical substances like poison could be a mutagen, or physical events like UV light or different kind of radiation could be also a mutagen. And we classify mutagens into two different categories. So let's say we have a person over here. A mutagen could be classified as endogenous. If it comes from inside this person's body and it's some mutagen that's...
Wait, nagkamali ako. Endogen.
So let's say we have a person over here. A mutagen could be classified as endogenous. If it comes from inside this person's body and it's some mutagen that's already found in the organism. But an exogenous mutation is one that comes from outside the affected organism, something that's from the external environment. Let's talk about ionizing radiation. What is ionizing radiation and how it changes your DNA? Ionizing radiation, which is energy that's given off by radioactive materials like phones, sun, stove, or even the light you're seeing on the screen is radiation. Ionizing radiations include ultraviolet rays, X-rays, or gamma rays, which makes sense, but what can this stuff do to you when it hits your body? Do that enough and all the high energy can cause chemical changes in our tissue? If ionizing radiation affects too many cells at once, or we absorb a bunch of over time, that's when we risk sickness, radiation poisoning, or eventually cancer. That happens when radiation changes how things fit together. It might knock off bits of our DNA, mess with its structure, or break one another both strands of DNA double helix.
DNA is constantly
pairing and multiplying itself. It builds with base pairs. Guanine always goes with adenine, and thymine always pairs with cytosine.
Sometimes, however, a chemical mutagen can cause a piece of the DNA link to be misplaced, or the wrong link to be added.
In this case, the DNA strand will do one of two things. Either continue to build off the incorrect link with effects that will be discovered later, or the sequence will discontinue because the links cannot fit together. This is a mutation.